Hello, my name is Fergal Waldron and I'm an Advanced Research Fellow at the University of Aberdeen. And I'm delighted here to be talking to you here as part of the MND Scotland LEARN event. And what I'm going to be talking about today is developing tests to improve access to ALS MND therapies. So what I will be talking about is how can we learn from cancer? How can we take some of those learnings with us into MND and ALS research? And I'll also be talking about a particular type of ALS called FUS-ALS, which is very rare and not very well known, but we think from some of our research here in Aberdeen with collaborators that FUS ALS, or that FUS as a mutation and as a protein may well be having effects in a wider range of ALS types other than strictly FUS ALS. So, two things that we should be learning really from cancer. One thing is that we got better at diagnosing cancer. We didn't necessarily get that much better at treating cancer. We do have better treatments than we had previously, but we've made huge leaps and bounds over the last 10 to 15 years. And this has primarily been achieved through diagnosing cancer earlier and being able to detect it earlier. So this is an infographic from Cancer Research UK, and it's showing that diagnosed at a later stage, that lung cancer has a survival rate of less than 1 in 10. Bowel cancer is very, very similar. It's 1 in 10, and breast cancer 3 in 10. So this is what is diagnosed at the latest stage. However, when it is diagnosed at the earliest stage, we have much better survival rates and much better prognosis for patients. So for lung cancer, the survival is about 6 in 10 if we pick it up early. Bowel cancer, very good, 9 in 10, and breast cancer, very good as well. So if you pick it up early, the prognosis is quite good. Also, one of the things that we need to learn from cancer, perhaps, is how do we use the therapies that we have more broadly. So this is a story about immunotherapies. So back in 2015, to treat lung cancer, researchers started using a drug, an immunomodulator called PD-1 inhibitor. And this is for treating lung cancer. So the cancer itself, it couldn't be seen by the immune system. However, when you use this PD-1 inhibitor, the cancer can then be seen by the immune system, and the cancer can be destroyed by it as well. But what was actually found to be the case is, so what was initially designed or thought would only really work and be effective for lung cancer has actually been effective for lots of other different types of cancer. So in effect, essentially, we've been able to use this treatment much more broadly across cancers than was initially thought. And we're able to do that by using paired biomarkers, so that we can look at how the effect we can look at the effect of the drug over time. So what can we learn from the success of cancer? First thing is that we need better tools for early and accurate detection. And the second thing, perhaps, is to ask whether existing therapies could be useful in a different setting if we use these new tools and these good biomarkers. So I'm going to start talking about FUS-ALS now. So this is a new snippet reporting on a modern ALS success story, I suppose, which was that an ASO, an antisense oligonucleotide treatment that was developed by Professor Neil Schneider in Columbia University with collaborators as well, has been shown to be effective in a small number of people with FUS-ALS. And this is the publication that's most recently come out about that. So one of the things that we need to think about when we're thinking about different therapies are what kinds of therapies can we use. So let's delve into what an ASO therapy is essentially. So your genes can be considered as recipes for making proteins that you're going to need in your daily life. And essentially, if you have a mutation, as some people do with SOD1 or with FUS in ALS or MND, that means that that recipe is not functional. But the ASO can function as like a sticky plaster or a sticky label, which then can correct the recipe as such. And that means that that can be used to target faulty genes that are causing the pathology or the disease. So here we see in a control, so this is brain post-mortem tissue of someone with FUS-ALS, and this is a FUS aggregate. And as you can see here, when they're treated with this ASO from the trial, from Neil Schneider's trial, the FUS aggregation itself is reduced dramatically. So this has been shown to be quite a successful therapy for FUS-ALS. So the FUS-ASO has only been tested in those with the FUS mutation. Now the question is, could it be useful for other people? Could it be useful for people with C9 ALS, SOD1 ALS? Could it be useful for people with sporadic ALS? So a few years ago, research from our group, we looked at ways of classifying pathology in C9 ALS. And actually, one of the things that we found, which was quite surprising to us at the time, is that we did find FUS aggregates, and we found that FUS aggregates in C9 ALS were a better discriminator, in fact, between ALS and controls than TDP43 aggregates, which are well known, or rather more well known than FUS aggregates in terms of association with ALS. So if FUS pathology exists in people who don't have a FUS mutation, perhaps we should be thinking about whether FUS therapies like the ASO I was talking about could be useful in those people too. So how do we find people who might benefit from this? So we need better tools to detect FUS accurately. So we need better biomarkers. So one of the things we do in our group, in collaborations with others, is we develop detection molecules called aptamers. So in biology, you might have, as you want to detect a protein, which is represented by this blue squiggle here as in the figure. And what you have is you have a red antibody that comes in and then detects an epitope, which is this small little box here. So that's quite specific for that epitope to detect the protein. And then you essentially stick a large visualization molecule onto the antibody so that you can see that under a microscope. Problem is, in neurodegenerative diseases, a lot of the proteins are aggregating, which means that rather than being nicely laid out in a string as above, they're aggregated and they're clumped, which means that the actual epitopes, the areas that the antibody is trying to detect, may well be hidden or difficult for it to find. So that means that the antibody can't essentially find the aggregate. So what we have is we've developed aptamers, which are much, much smaller than antibodies and actually detect structural epitopes, which aren't necessarily hidden in the clump. And because they're much smaller, we can actually attach many of these aptamers around the structure of the protein aggregate. So just to compare and show you how this would work, when you use your visualization molecule as well, using the aptamer approach, sometimes we can detect molecules that you can't detect using the other approach. And in cases where you might be able to detect the aggregates using either approach, the resolution that is allowed with the aptamers is much, much better. So we started thinking about let's make a FUS aptamer, and we did this in collaboration with our colleagues Gian Gaetano Tartaglia and Elsa Zacco in Italy. And these are designed computationally. And this is a representation of what a FUS aggregate or a FUS protein looks like, I should say. And here is a little sort of kind of a little nook in the protein as it forms this little structure. And this is where the FUS aptamer actually detects the aggregate. So once we had created the actual aptamers themselves, the question was, do they work, and how can we use them? So in collaboration with Neil Schneider from New York, who was involved with the FUS ASO trial, we were able to get some post-mortem tissue from people with FUS ALS, some of whom actually were involved in the trial itself. So we have some people who were treated with the ASO. So here we see, looking at the antibody, what FUS looks like in its pathological state. So in the control brain tissue here, we see a normal homogeneous nuclear distribution. So it's kind of this sort of egg-like structure, essentially like egg yolk, which is in the nucleus. But in this person who had FUS ALS and wasn't treated, this is, you can see the cytoplasmic aggregation. So this is what FUS pathology essentially looks like. Also, using the antibody, we can see that in the ASO treated individual here, or the first ASO individual treated, that there was no normal or abnormal staining whatsoever. So what had happened is the ASO had essentially gotten rid of all FUS, normal and abnormal. So it had a huge effect in reducing FUS overall. This second individual here was someone who was treated with the ASO, but stopped taking the ASO sometime before the end of the trial. So what had actually happened here is that the aggregates, which would have been reduced upon ASO treatment, they appear to have come back to some degree, having stopped taking the treatment. So we have developed two aptamers, and I'll show you now how they compare to the traditional antibody approach. So the first question that we're asking is, what's the sensitivity of these aptamers? So can they see the detail that the antibody can see when we compare the two approaches? And we find that they actually can. So that's good. So we are picking up the nuclear and the cytoplasmic aggregation that you can see with the antibody using both of the aptamers, and that's pretty widespread as well. So the next question then is, does it have good specificity? So the point of the matter is, can it ignore things that are not relevant, that we don't particularly want to be seeing, or that we don't want to be focusing on? And in fact, we find that it does have improved specificity compared to the antibody. So whereas the antibody picks up a certain amount of FUS staining in our FUS aggregation in the control individuals, we see no pathological staining whatsoever when we use either of the aptamers. Also, similarly, a question that we would want to ask is, can it see more? Can either of the FUS aptamers be used to see more pathological features than the antibody can actually reveal? And we find as well that actually it is the case that that's true here in this case. So if you look here at the individual who had stopped taking the ASO, so this is the PR521L individual, some mild pathology is observed there, which isn't actually really visible with the antibody. So our FUS aptamer has equivalent sensitivity to antibodies that are available. It's got improved specificity, and it can see more than the antibody can as well. So what are the next steps that we're thinking about? So firstly, we're thinking about can we develop a blood test so that we can use our aptamers for accurate detection of FUS pathology? And also start thinking as well, not just in terms of biomarkers and developing aptamers for biomarker and monitoring purposes, but also we can use aptamers for the targeted treatment of FUS pathology as well. So that's something that we are going to develop as well. And because we see FUS pathology in a wider range of ALS subtypes than is widely appreciated, we think that this is something that could be generally applicable across ALS. So, in summary, when we're learning from cancer, perhaps we should consider these crucial lessons. Firstly, where cancer made huge progress was in early detection. So what we need to be doing, and I think this is generally acknowledged across the ALS field, is that we need to be detecting ALS as early as possible so that we can either stratify people for the right treatment or provide whatever treatment is appropriate at that time and get people into clinical trials as soon as possible. And secondly, can we maximize useful therapies? So we have therapies like the FUS-ASO and tofersen, and the question is, with the FUS-ASO in this particular example, is it possible that this therapy could be useful across a wider range of ALS subtypes, including sporadic ALS, and we have more work to do on that to see if that's the case. So that just leaves me to acknowledge the people in our team. This course is led by Professor Jenna Gregory and also our collaborative network, NHS Grampian, and in Columbia University, IIT in Genoa, and also in the University of Edinburgh. And thank you very much to MND Scotland for their great work and for the opportunity to speak at this event. Thank you very much.